So we'll now move to our next speaker, who is Dr. Michel-Yves Mistou, and he is head of the Foodborne Pathogens Department of the Laboratory for Food Safety in ANSES. Dr. Michel has been with ANSES, which is the French Agency for Food Environment, Occupational and Health Safety, as head of the Foodborne Pathogens Department of the Laboratory for Food Safety since 2014. He leads a change management strategy towards the implementation of whole genome sequence technology for research, reference and surveillance activities. He's also responsible for the GAMR team in charge of bioinformatic development. Michel will share with us the genomic projects in ANSES Laboratory for Food Safety. Thank you.
Thank you, Patricia. And thank you also, Jürgen, for organizing this wonderful seminar, which gave me the opportunity to present our work. So I will start my presentation with an overview of the organization of the Laboratory for Food Safety, which is headed by Laurent Lalou, and which is composed of three different departments. A department focusing on the sea products, and which is located in Boulogne-sur-Mer in France, so the first fishing port in France. The Department of Chemistry, whose activity is on heavy metal, pesticide, marine biotoxins, and the Foodborne Pathogen Department. Both these departments are located in Maison Alfort. And the Laboratory for Food Safety, it's a staff of around 140 people. Now I will focus on the Foodborne Pathogen Department. The staff is around 80 people, 2/3 technicians, 1/3 scientists, and it welcomes every year around 15 to 20 master, PhD students and postdoc people. The Foodborne Pathogen Department, it is made of 6 different entities, which are focusing on foodborne pathogens, so enteric viruses units, Salmonella and Listeria units, and the toxin-producing bacteria units, which works on Staphylococcus, Bacillus cereus, and Clostridium perfringens. We also have in the department a national platform, ANSES national platform, which provides services on high-throughput PCR. So they have a microfluidic instrument facility there, and they are also doing a quite intensive research on E. coli tech. We have then the official laboratory, which is an official laboratory which is dedicated to investigation of foodborne outbreak in the Paris area. So they are doing foodborne detection in food samples. And then the 6th entity is the Genomic and Modelisation unit, but I will come back later on the role of this unit, which is of course pivotal for all our WGS activities. Importantly, in the department, we have 4 national reference laboratory mandates on Listeria, Staphylococcus, Salmonella, and enteric viruses, and 2 EURL mandates on Listeria and Staphylococcus. And also very important is that the department maintain and continuously enrich its collection of foodborne pathogens on Salmonella, Listeria, and all the pathogens we are working on. And it is important because many of our genomics programs try to take advantage of this collection, of this rich collection of bacteria, essentially. What are the typology of activity in the department? So our main business is detection, typing, and characterization of foodborne pathogens, and this is performed in many different contexts, like surveillance, foodborne outbreak investigations, which are usually coordinated by the French Public Health Agency, Santé Publique France, and which is usually performed in close collaboration with the CNR, the National Reference Centers, which are, in our case, located at the Institut Pasteur. We provide also scientific and technical support to ANSES different divisions, to national and European public health authorities, EFSA, for example. We have also a lot of activities around the coordination of laboratories networks, and we provide technical support. We organize proficiency trials, and we also organize training courses for our network of laboratories. And we have research projects which have as main objective the development of innovative methods in our activities. And of course, we are well aware now since many years that implementation of whole genome sequencing approaches and technology is crucial and mandatory, I would say also, if we want to maintain our position and if we want to conserve our scientific credibility in our activities. So it is easy to say that okay, WGS is important, but how to get there and how to pass from the conventional method to the unique technical platform that WGS is, because we have to meet many different challenges in terms of expertise, staff training, financial resources, IT infrastructures, regulatory issues, and so on. And so we have to find our specific way to do that. And I think that we have there are many different contexts, many different culture, many different histories in the labs. And so I will just describe you now our way to go to WGS, which is not a universal way, but how we proceed. So our strategic line was first to choose to externalize the sequencing activity, because at the time when we started, we simply did not have the resources to perform at home the sequencing, and so we chose to go through a public tender and to find a provider to do sequencing for us. And we choose to concentrate rather on data analysis, and so to build internally our data analysis expertise, and so to build a bioinformatic team doing this for us. And so it means hire competent people, build IT infrastructures, develop in-house pipeline, and so on, but I will come back later on this. And also to try to develop genomic research project, because I think I thought it was the only way to go into this new field. It was to learn while doing. So the important point was to build this expertise, and for this we have created in 2016 the Genome Analysis, Modelisation and Risk team, that is called GAMR team, with the following objectives. The main objectives were to develop the IT infrastructure, develop in-house data analysis pipelines, organize, and this was a very important point. If we want to spread the skills and the expertise among all the scientific staff, you have to train the people, and this was one of the very important missions of the unit. Of course, it was supposed to provide full assistance to genomic project developed in the department, also to animate and to organize seminars and thematic days around the genomics, and of course, and this is the last but not the least, develop its own research project, because the people you are hire in this team, they are young people and they need to be creative. They want to have an interesting job, and they just not only want to help others, but they also want to make their own.
What does scientific animation mean? Is it a special French thing?
Yes, it should be. It is organization of seminars, essentially. Okay. So this is the actual composition of the GAMR team. So it's around a dozen of people and 3 permanent people among them: bioinformatician, genomic specialist, and informatician. So the first mission was to set up a GNU and a Linux-based environment around a Windows-based environment, which is the case at ANSES. And so this IT infrastructure allowed an easy local access to calculation and storage resources. And through training session, many of the scientific staff, microbiologists and molecular biology specialists, learn how to access data and run analysis. The second point was to develop our own analytical pipelines for WGS data. And so this pipeline was developed. It allows, it performs data quality control, data curation, variant calling, assembly, typing, annotation, and so on. And this is really the entry door of all the sequence we are doing in the lab. So they are running through this pipeline that we know very well, its limit, but also its advantages. And the third mission was to develop a local database to ensure the easy and secure access to our genomic data, allowing storage, backup, and traceability, and development also of a web interface for easy querying of the data, and also querying of the analysis performed through the artwork pipeline. So with all these developments, it was possible to start to perform a genomic project. And so I will now go through 3 different projects that I choose among the 20 or so which are actually running. And the first one is in fact, it's a good example of the first example that given by Jürgen, and concerning what WGS can bring to source attribution problems. So this project is called Derby Clone. It was financed by the Ministry of Agriculture, and it financed a PhD thesis, in fact. And it's about Salmonella derby, which is one of the serovar of Salmonella, which is among the top 5 clinical serovar in France. The first serovar isolated in pork meat in Europe. This is data from EFSA. And unfortunately, concerning this Salmonella derby, but fortunately for people doing WGS, you cannot discriminate between isolates of Salmonella derby through the classical conventional PFG method, and so it means that you cannot trace back source of contamination with this serovar. So to gain insight into the epidemiology of Salmonella derby, we have sequenced around 140 animal and food strains from our own collection, which were representative of the production sectors and of the different geographical areas of sampling. And to go fast, what we discovered with the WGS analysis is that behind one serovar, you got 6 different genetic clades, and which were highly specifically associated to a specific host. So you have clades which are associated to pork, clade associated to fowl, and another one which is not extremely frequent, but which is also associated to pork, even if it is quite genetically different from the other ones. So these results were recently published, and what we have done next is to add into this picture human isolates. And for this, we have contacted our good friends at the Pasteur Institute from the National Reference Center of Salmonella, and who provide us 302 human clinical isolates sequences. And on the left, this is a phylogenetic tree of food and animal strains, and thereof you add the humans in blue on the external circle. And what we found is that 98% of the human isolates are linked to the genetic clades associated to the pork sector. So this provides a clear extrainable information. So it means that if the risk manager wants to act on Salmonella derby, he must focus and target its action on the food sector, and it's not necessary to bother the fowl sector. So it is a clear, I think, example of what WGS can bring to the public health safety. The next example I will give about our WGS activity concern a recent foodborne outbreak investigation, which was carried out at the European level. It was in the last July, multi-country outbreak of Listeria monocytogenes, ST6, 47 human cases, essentially in Finland and Austria, and 9 deaths associated. The outbreak was coordinated by ECDC and EFSA, and our EURL on Listeria monocytogenes was in charge of trying to get some food samples from its NRL networks. And so for this, the EURL interviewed its laboratory network to select, receive, sequence, and analyze 29 isolates, which were genetically close from the human cases. What we found is that we got some strong microbiological links between the selected isolates and the non-human isolates. Human isolates are in red on this phylogenetic tree. And this activity is, I think it's quite a good example of a good coordination between public health agency with a good management of a laboratory network, and then after that, correct confrontation between epidemiological information and WGS analysis can really perform very well and provide a good investigation. And at the end, the conclusion was the contaminated food was frozen corn, which comes from a freezing plant in Hungary, and of course, whose activity have been halted since. The third and last example I will give you is about a more exotic foodborne pathogen, which is Clostridium perfringens. But it has not to be neglected because we have always, with following the EFSA figures, 15,000 human cases in EU per year. It ranks fourth in the toxin-producing bacteria associated with food poisoning outbreak. And at the moment, the genetic diversity of C. perfringens is very poorly characterized. And at the moment, there is no typing method which are relevant for investigation. So what we have done, we have chosen among our collection of Clostridium perfringens a number of strains which were also representative of what happened in the last 10 years. And we performed genomic analysis of these strains. What we found is that these foodborne outbreak associated strains can be divided along two different clades, which are in fact associated with the presence of the CPE genes, which means Clostridium perfringens enterotoxin, and which was until recently considered as the main determinant of the enteropathogenicity of Clostridium perfringens. But as you can see here, all these strains have been isolated from foodborne outbreak, but they do not possess a CPE gene. So I think this study revealed new feature for Clostridium perfringens food poisoning, but it also raised new questions. For example, that food poisoning is not always associated with the presence of the CPE gene. So are there other toxins involved in the enteropathogenicity of the species? We also observed, I didn't get into the detail here, but that there are a very unexpected diversity of the CPE locus. So we don't know yet at the moment what is the impact of this genetic diversity on toxin production. We also found that 25% of the foodborne poisoning outbreak are associated with very heterogeneous contamination by Clostridium perfringens, meaning that in the same food sample, you have very different isolates of CP. And of course, this is important when you want to know exactly what is the cause of the disease. And it also raised the question of, is the standardized detection method, which there is even a norm, ISO norm on it, is really relevant for foodborne outbreak investigation with this pathogen. And finally, this work also allowed us to develop a multiplex PCR method, which targeted 19 toxin genes. So it allows also for rapid typing of isolates, which was not previously available. So this is the last project I wanted to describe in detail. So these are the people which were involved in these three projects. There are many others. I did not want to go into. So we are involved in several projects in the framework of the Compare project that was described by Jürgen and Roger. We are also involved in one else EGP project, which is under ANSES coordination for a total fund of 90 million euros. And especially I would like to mention the RADAR project, which is about new models for the dissemination of antimicrobial resistance. And so we are working on how to fit this model with genomic data, and especially the mobilome data, so the data related to plasmids. An interesting project also on Bacillus thuringiensis, and does it represent an hazard? Because as you maybe know, Bacillus thuringiensis is used as a pesticide, which is authorized in organic food products. But all the bacillus, they are very close from each other. And for example, Bacillus thuringiensis is very closely related to Bacillus cereus, which is a foodborne pathogen. So we are investigating this potential hazard in organic food product. We have also a project on the modeling and quantitative risk assessment, because in risk assessment usually neglects the fact that within a species you have, as we have seen, a lot of genetic diversity, and when you have genetic diversity, you can have pathogenicity diversity. And this is not really taken into account at the moment. And we have a very nice paper on because it is now known, for example, for Listeria monocytogenes, that you have hypervirulent strains, medium virulent strains, and strain with very low virulence, meaning that you never find it or find them in clinical cases. And this was not until now taken into account in risk assessment, and I think it should definitely be. And so we have a paper trying to remodel this aspect, and also other more fundamental research which are driven by the GAMR unit. So this is our acknowledgement and the different collaboration we set up. And before to thank everybody, I wanted to give you a last message. So ANSES is organizing conjointly with BFR and DTU Food and NIFDS a joint conference in Paris next March, and which is entitled Impact of Whole Genome Sequencing of Foodborne Pathogens on Public Health Protection. Thank you for your attention.
Please join me in thanking. Thank you. And consistent with earlier, we'll save all of the questions until the end, and hopefully we have time. And I'll just make the comment as well that if the speakers are willing, we will provide as much material as possible as handouts and also contact details so you can follow up with the speakers if you have further questions.